So thank you for joining us today. My name's Emily. I'm going to walk you through UniProt Proteomics Scientists today. So I can obviously give you an introduction to UniProt and some tools that you might find useful when you look at large-scale protein data. But we have many training tools online, as Anna said, and she's going to be sending you a few links for those. And I'm also going to be highlighting a few more useful webinars for you at the end of this session. I'm a scientific curator at the UniProt database, which means that I read published information and articles and add that data to the database in a protein-centric way. And I'll tell you what that means shortly. But I'm also joined by John and Swati, who are part of our development team, and they'll be able to answer questions that I don't know, basically, about the background behind the database and how it's structured and put together and how the data is processed. So our aims for today, I'm just going to move my little sharing bar out of the way. So our aims for today are to learn, for me to walk you through some of the website tools that we have available on UniProt that you might find useful in helping you to analyze your data. And then I'm going to briefly introduce you to the API and how to access some of the proteomics peptide sequences and data that we have available through there. So hopefully you'll be able to access the website on your own accord tomorrow, and you'll have an idea about where to find these tools and how to use them in your own pipeline. And you'll also have an idea of where all our documentation from our API is, how it's structured, and how you can retrieve some information from there, either using programming methods or a multi-query search function that I'm going to show you later. So, who are we? We are UniProt. We aim to provide a comprehensive, high quality, and publicly accessible resource that's always free. We provide protein sequence and function information. So we store primary sequences as well as isoforms, and we curate and collect physiological protein function, which means we will focus on wild type protein function in a wild type environment. So always it's normal function. And this will include subcell localization, any pathways it's involved in, whether it's involved in variants and disease and how that might change its functional pathway, any interactions and reactions, as well as any other information we can capture and annotate. We also annotate the amino acid sequence, which is called our features, with certain domains, post-translational modifications, mutants, variants, and also much more. We also have structural information, such as topology and access to 3D structures, including papers and AlphaFold. And we have data analysis tools such as BLAST and multiple sequence alignments, which I'm going to talk about later. UniProt is brought to you by the UniProt team, who I'll have a slide on all their names later. But we're spread across 3 sites around the world, at EMBL-EBI in Cambridge, UK, at PIR in the USA, and CIV in Switzerland. So briefly, I'm going to talk about how our database is structured, and it's broadly split into 2 sides. So I'm talking about UniProt knowledge base right now, which is where all our proteins are stored. And we have 2 sides. So we have the unreviewed and the reviewed side. Unreviewed was originally called Tremble because it was stored at EMBL-EBI. And then our reviewed side was called SwissProt because it was stored in Switzerland. And how the database is structured is all our unreviewed entries are curated using annotation rules. I'm sorry, I'm going to have to just get rid of this bar because it's annoying. Hide, close the meeting controls. Lovely. So our unreviewed side of the database is where our direct sequence submissions go in, and we store direct sequence data from cross-reference repositories. These sequences are then computationally annotated and information added to them based on those curation-led rules. Those rules are put together and curated and inferred by curators such as myself, and we all tend to have a specialism in that particular area or the particular enzymes or that particular species when we curate these rules. And that information goes into the entries on a protein-centric basis. So each page within UniProt is a protein, and in the unreviewed side of UniProt, it will be there will be an entry for each isoform. So if a protein has 3 isoforms, there will be 3 entries associated with that particular protein. When we bring a protein in from our unreviewed side to our reviewed side, A curator, such as myself, will look at those protein entries. We will double check the sequence or the annotations or the data that has been brought in automatically via our previously mentioned resources. And then we will also go through the current scientific publications and add data from those. We will pick the canonical sequence for the protein, and the isoforms that we have associated with that will be merged into it. So in our reviewed side of the database, we have one entry for each protein, and the isoform information is available within that entry. The sequences are then manually reviewed and annotated as well. So these are the amino acid sequences. You will get amino acid residue specifically annotated if the information is there. And we also do that for isoforms, and that information is available as well. So say the isoforms are grouped per canonical sequence. And throughout the database, you can see this by these little icons here. We have a white or a grey icon for our unreviewed entries and a gold starred icon for our reviewed entries. So in today's webinar, the things I'll be covering briefly are our proteome service. We'll be looking at how to search the database via peptides. I'll be looking at our ID mapping service and also how to access the data programmatically via the API. If you're unfamiliar with UniProt, as a web page or as a resource, we do have a walkthrough at the UniProt website and how we annotate data webinar, which I'll mention later on one of my final slides, which you will probably find really useful. So I'm going to briefly just talk about how you would download a specific protein sequence. So if you navigate to your protein of interest, in this case, I'm looking at PALS1 in human, you can access all the sequence information for this protein in the sequence and isoform section on the left. So on the left of the page, it acts like a tab bar. And if you click sequence and isoforms, it will jump to the sequence and isoforms section, which looks a bit like this. PALS has two isoforms. So first it will list the first isoform, and then it will list the second one. And you can align the two isoforms if you want. You have a bar here of different tools that you can use. You can add it to basket for analysis later. You can highlight certain domains, etc., etc. But if you just want to download the faster sequence for this particular protein, you can click download, and you will get a drop-down box here. When you click download, you get several options to download in different formats. If you just want the faster sequence, it will be one of the faster options you'll select. Canonical means you're just downloading the canonical amino acid sequence. If a particular protein has isoforms, you can download the canonical and its isoforms. And this is the file you will get, and it will download as a text file. So that's how to do a simple download of an amino acid sequence from any protein of interest in the database based on the website. You can also do this in the API as well. So if you're looking for your proteome of interest, so if you want your reference proteome to search against your mass spec data, this is where you find it. On our home page, we have a big red box in our resources section called species proteomes. And if you click that, it will give you a rather extensive list of reference proteomes that we have. I've selected human for you to have a look at because human is what I specialize in. And it will tell you how many proteins are in this proteome, the current gene count, its specific ID, which you should always include in publications when searching against a proteome, the taxonomy it's associated with, the link to its genome assembly, its statistics associated with the reference proteome, and then a brief summary of the species itself. When you scroll down the page, it will give you several options to download. You can download it either a selection of chromosomes or just a specific chromosome, or you can download it all. So if you click download, you can again select either faster canonical or faster canonical and its isoforms, and then you download that traditionally in a compressed file, which you can then use for further analysis down the line. So our retrieve ID mapping service I found particularly useful when I was working in the lab. This service allows you to swap or compare different IDs between an ID of interest and compare it or transfer it to a UniProt session ID. So the example I've used here is gene IDs, and I've converted them to UniProt session IDs. So why might you do this? If you had a load of data come off your mass spec and they're UniProt accessions, but your further analysis pipeline uses gene IDs, you need to convert those, and you need to be able to convert them in bulk. So you could copy or paste a link into our ID mapping service, or you can use a text file as well. So on our homepage, we have a search with lists, map ID, or we have an ID mapping tab at the very, very top of our web page that's always there. And it will open a page like what you see on the left here. And I've copied some gene names in, but I say you can also upload a text file. You can restrict it by taxonomy. If you don't restrict by taxonomy and you put the beta-catenin gene name in, you're going to get mouse, rat, zebrafish, basically everything we have in the database. So if you want something specific to a specific species you're working in, make sure you restrict by taxonomy. I've inputted 7 IDs. I've told it I've put gene names in and I want UniProt accession IDs out. And then you can name your job. So I've just called it ID mapping example. When you click go, it will take you to your toolbox and it will say, we're running an ID mapping job for you for this on that date. And it will be queued, then running, and then completed. And when you click completed, it will tell you your results. So this is what a results table looks like in UniProt, and they're pretty similar depending on what you search, but you get a table like this with different columns, and you can customize your columns, which I'll come back to in a second. I filtered this table by our reviewed entries. So you see our little gold box because I only want manually reviewed entries here. But you don't have to. You can have the full kind of results if you want from our unreviewed section. And as you see, I've got 7 IDs that I put in, and I've got 7 results that I put out. And that's what my current results table looks like. So let's say you've got gene names and you've converted them to UniProt IDs and you want to use them IDs in a downstream piece of software for some further analysis. So you've put in, let's say, 3,000 gene names and you want 3,000 UniProt IDs out, and you don't want to be copy and pasting that. So what you can do is on your results table, which looks slightly different now, but it's the same table. This is that table. I've clicked customize columns, and all I've done is I've deleted all the options. And that gives you the basic default for that results table. So you see I've got the gene names I put in here. I've got the accession IDs, the resources given me out here. And then I've clicked download. I just want a simple list format. Click no so I can see what I'm looking at, and that's the readout it's given me. Okay, so if you want to put a text file in of just gene names and get an accession ID out of just accessions, that's how you do it. You just customize your results table, delete all the other information that you don't want to download, and it will give you a simple text box like this. Bear this in mind when you're exploring the UniProt website, because every set of results tables will have a customized columns option, and it will affect what you can download. So let's say you put these gene names in and it gave you a set of entries, let's say 7 entries that you got these results from your mass spec, you want to know what they do. If you click customize columns and say, I want to see the function and its interactions, you can then see those results next to their gene names in this results table. You can click download and download that information. So any results table in UniProt, you can customize its columns, select exactly what you want to see, and then download it. But the principle here is just to give you a simple idea of how you can convert one ID to another. But the idea of customized columns allows you to download and view anything you find of interest in the UniProt website. So similarly, in peptide search, if you go on the home page, we have a purple box that's named Search Peptides, and we also have a tab of peptide search at the top of the web page that's always there as well. So you can use this to search peptides against a database to find proteins of interest that these peptides match to. So we have a similar box to the ID mapping service that you can copy and paste your peptides into. You can also upload a text file. You can restrict by taxonomy, so you don't have to. You can name your job. Here I've search reviewed entries only. Again, you don't have to, but I have, and that's why I have only got 6 results. So if you were to run this as an example, you get a few more. You click run peptide search. It searches the whole database for these particular peptides and what they map to. And this is the results table you get. You can search any number of peptides here, and it will tell you which peptide maps to which amino acid positions on which protein. So in this case, these peptides all match to GSK3 beta in a set of different organisms. So I've got frog, I've got mouse, I've got rat, I've got human, and they all match to these peptide positions. And I say you can customize columns, which is how I've got it to show me protein names instead of accession IDs or gene names. And then you can download that information if you want. Or the tabs on the left here of any results table that we have, you can filter by. So if you only wanted to see human, you could. If you only wanted to see entries of the 3D structure information, you could do that too. And then you could download all that information if you wanted to. So that's how you access peptide data via the website, and we'll be looking at that in the API shortly too. And now I'm going to introduce another bit of proteomics data that we have in UniProt. This has been around about a year now. So this project is called the PTM Exchange Project. It is a collaboration between UniProt, Pride, Peptide Atlas, and the University of Liverpool. And it takes a set of data sets from the Proteome Exchange that are then filtered with particular methods, sample types, and robustness of study. It's then analysed and compared to Peptide Atlas, and it's reanalyzed in a context that gives a scoring for each post-translational modification before the results come to UniProt. And what that means is each PTM that is mapped by this proteomics analysis has a confidence score, and we've done that according to gold, silver, and bronze categories. The confidence score is based on the false localization rate of the data. It's calculated at a modification level within each data set. So if you've got a modification that's below a 5% FLR and it's seen in one or more data sets, it will be given a bronze classification. We have just short of 6,000 for our rice proteins here for that. So that's just short of 6,000 sites, not proteins. To get a silver category, that PTM will be seen in one data set at less than 1% FLR, and then to get a gold, it's seen at less than 1% FLR in more than one data set. And you can see we've got a few less of the gold because it's a more stringent category. So how can you view this data? So this is just in a protein entry page, and if you want to explore this yourself, this is the accession ID that you need. You go down to the PTM and processing section on the left tab section, and it opens a section a bit like this. You get a table and a graphic at the top. The gold bar along here is the protein chain, and the little notches are all the little PTMs that we've got data for. I filtered this data in this table for modified residue large scale because the data is from large scale analysis, proteomics data. And the source is a PTM Exchange project, but you don't have to have these filters on. I've just put them on for ease of showing you where the data is. And then it will list where you've got the modifications at. So these are the amino acid residue numbers, the modification type, its confidence score, bronze, silver, and gold. And then the grey tab here tells you where that information has come from. So if you were to expand one of these rows, it will tell you the further information, and you click, and it also highlights it on this table, which is difficult to see when it's sort of the PTM because the bar is quite small, but if it was a bigger domain, you'd see that. And then you click the combined sources evidence tag, and it tells you this information is from experimental evidence that we found in Proteome Exchange, that we've also computationally analyzed. This particular PTM has got a confidence score of gold, and there'll be further information down on the scrolly bar that tells you where it's from. So it will give you all the PRIDE identifiers for the data sets as well. Throughout UniProt, we have many of these little information eyes. Please make use of these and click them. They would take you to the most relevant help pages. And in the case of this project, it will tell you all the methods of how data like this is calculated and reanalyzed and displayed. So please use them. And the same with our help tabs. It will send you to the nearest help page that's most useful to you and what you're viewing at the moment. So going forward, if you want to view it in a different way, you can click the feature viewer option at the top of the entry page. And when you click the feature viewer, it gives you a table, a graphical view of a table. where you've got along the top your string of amino acids from N to C terminus, left to right. And on that table, you've got a set of tracks, and each track is a different type of information that we've mapped them amino acid residues. And for the PTM Exchange project, the data is found in 2 of these tracks. So we've got a PTM track and we've got a proteomics track. You can see we've got many other tracks as well going up here, but I'm just going to talk about these 2 today. So if you click the PTM track, the PTM Exchange data is shown under modified residue large scale data. And then each PTM will be shown as a little triangle that's color coded according to bronze, silver, or gold. And then when you click on the triangle, it will give you the information for that particular modification. So as you can see here, it's a amino acid 150, it's a phosphoserine, it's got a bronze confidence score, and it's been found in these data sets here. And if you click the PRIDE link, it takes you to PRIDE in those data sets. If you want to see this in a peptide context, you can click the Proteomics tab, and that opens up and shows you unique or non-unique peptides, and then any peptides that have been mapped containing a PTM. So in this case, your peptide is a little kind of pink bar here. And then within that peptide, that navy line is your PTM. And when you click on those little graphics for your PTM containing peptide, it will tell you the amino acid residues that that peptide is spanning. The amino acid residue that's modified, so we've got phosphoserine at 189 here. Its peptide form notation for that particular modification. It's mapped to this proteome and that proteome ID, and the data sets that they come from. And we've also got peptide atlas links here as well, if you really want to dig down into that data and see the spectra as well. So now I'm going to briefly introduce the Proteins API. This will allow you programmatic access to the database. You don't need to be able to program to use it. I don't program and I can use it. I'm going to walk you through the 2 different ways you can use the API and how it's going to help you access this data. If you want to learn more about the API, in the autumn, we run a programmatic access to UniProt, and that combines Python and the website together as well. That's a recorded webinar that we did last, I want to say November. So I'll show you the link to that later that you might find useful. But on the home page for now, if you scroll down, you get to a UniProt data section, and you want to click the big grey box saying programmatic access. We also have a GitHub link if anyone's interested there. The programmatic page, access page, will give you a page that looks like this. We have a set of APIs, and they all come with their own documentation. But this is the one we're going to be looking at today. And if you click on the documentation, there's a published paper there that explains how the API is put together and what you can access and how to use it. And I'm going to give you a very brief walkthrough. So it's important that you understand how the API is structured. It allows you to access data from external resources and also how to filter that data out. So you can, as an example, filter for specific external databases, but you're looking for variant information. So you might only want data from Cosmic and not from other sources. So the API gives you that functionality. It's multi-query, so you can search by disease or OMIM ID within a certain taxonomy, or within a certain set of variants, or across a certain part of the genome, as an example. And you can retrieve data for separate canonical and isoform entries as well. So this is briefly how the API is structured. In the middle, you have all your protein information and protein entries, including your isoforms. Our coordinates service tells you where that's all mapped to on the genome. Our reference proteomes are what I spoke about earlier and showed you how to download. Our proteomics section shows you all our large-scale data sources and mass spec derived peptide sequencing, and we also have those mapped to isoforms. The variant service shows you all our large-scale data sources for protein variants mapped to disease. Our features function, which means the amino acid string features, will map all the domains in different regions and binding sites to the protein's amino acids. The antigen sequence takes information from the human protein atlas. So if there's a particular part of your protein you want to bind an antibody to, or you want to find an antibody for a particular protein, there's that service that you'd find useful. And then there's our taxonomy service, which stores all our classification of organisms as well. So you can access all these through the API and search by various methods. And this is what it looks like. So this is the multi-query search format in this gray box. And each one of these tabs on the left will open up a slightly different multi-query search function. So in variation, you'll be able to search for slightly different things compared to what you will in proteomics. So if you want to go and explore these, my advice would be figure out where in the website, which section of the website or an entry page is the information you're interested in, and then go to that section of the API and figure out how best to filter for that in the API. And I say each format, each query is different between each of these sections, so you'll get used to using what is good for you in your analysis. So I'm going to introduce you to both of the proteomics sections today. So the proteomics section will open up a form like this, and I've done a really simple search for an accession ID because I want to know what peptides are unique to that protein. So I've got the UniProt accession ID for that protein. I've hit try it out, and the first thing you're going to get as part of your results is a piece of sample code. So you get, for those of you that don't program, you get a request URL that will allow you to, if you keep a hold of that, it will allow you to download those results in future, and you won't have to rerun the search. For those of you that do program, we have a series of different code examples. So curl, Perl, Python, Ruby, Java, and R. This is the Python response body. And then this is the R, and you can save that for yourself to recall that search, or you can chop and change it to fit into a pipeline of interest. It basically gives you an idea of the code you'd need to run this query at home from your own terminal. And then going down the page a bit further, you're going to get your response body, which is your results section. Now, for those of you that are familiar with programming, yes, I've copied and pasted quite a few bits and missed a few of these little bars out, okay? But I've done that to fit it all on one page, so I can show you how the results are kind of ordered and formatted. So I queried one single protein here, and it said, right, your first peptide starts at amino acid 123 and ends at 141. That information has come and is mapped to that particular proteome with that ID. The data for that has come from Peptide Atlas, and here's its URL if you want to go check that data out. There are also a couple more sources for that down here. So if we've got multiple sources for a peptide, you'll have multiple source information. And then below that, you get to your sequence and uniqueness. So it will feed you back what that peptide sequence is. And then uniqueness being, if I put that peptide into the search, is it only going to pop up that one protein, or does it map to more than one protein? If it only maps to one protein, it's unique. If it maps to more than one protein, it's not unique. And if it's not unique, that will be false. Okay. If you put in a simple query, accession, like I've done here, accession ID P41841, and no other filter, it's going to spit every single peptide possible back at you. But you can then filter it. So you can filter it by human, you can filter it by where that data comes from, you can filter it by whether it's unique or not. So this is what I mean by multi-search. You can stack your searches up to get more specific results back for you. So what happens if I've got the opposite? What happens if I've got a couple of peptides and I don't know where they come from? So this is like the reverse of that search. So we're still in the proteomics section of the API. I've got rid of the accession ID here, but I put my peptides in the peptide search. If you've got more than one peptide, that's not a problem. You just put a comma between the two of them. And I've also told it I want these peptides to be unique to that protein. And then I've clicked try it out. Your results are going to look slightly different this time. It's going to give you back a protein ID that's uniquely mapping to, and then the entry name. So we're back looking at GSK3 beta in human. It's giving you a protein amino acid sequence. That's for the whole protein. And then it said, I'm in human here, tax ID. And then further down, it said, your first peptide will map to amino acid 329 and finish at 354. That information is from Peptide Atlas. Here's the URL if you want to go check that out. I've given you the sequence of your peptide back, and it's unique to that particular protein. Now, if you've put in here more than one peptide like I have, the next section will be your next peptide maps to 151 and ends at 182, and then the source and its uniqueness, etc., etc. If you put two peptides in here and they map to different proteins, your results will look like the first bit here, and then underneath here, it will say a different protein name, a different entry name, a different amino acid sequence, etc., etc. And it will always feed it back in the order that you give it the search peptides. And you can obviously download all this information as well. So looking at back to our PTM exchange data, I said I'd show you how to get this information out of the API. So the Proteomics PTM section, you can just, I've just searched by an accession ID. So this is to get all modified peptides that were found within that protein. And that's the same protein that we were looking at earlier in our examples. It's a rice protein. And then you can filter by confidence score. So you can even say, I only want gold confidence scores PTMs back from him, or I want silver. And then you click try it out. The results you're going to get here are where your peptide start and ends again. And then it will tell you the proteome it's mapped to, the peptide sequence that it's pulled out and whether it's unique or not, and then the type of modification that's within that modified peptide. So you've got, it's a phosphorylation in this case. It's at position 15 within that peptide string. And the source of the data is the PTM exchange. And then below that, it will tell you it's from this particular data set, this PXD in Pride. That data set is associated with that PubMed ID. It's got a confidence score of gold. It's got PSM scores, etc., etc. So this information is specific to that data set. So if this particular peptide is found in more than one data set, there's going to be another PXD down here below this set of data with PXD X, Y, and Z with a different PubMed ID, a different confidence, well, it'll have the same confidence score because it's the confidence scores at the PTM level, but then it will have slightly different statistics because the statistics are specific to the data set, not the PTM. So you could well have like 3 or 4 data sets down here. If there's more than one peptide for that particular protein that's modified, once it's finished giving you back all the statistics, it will start with the next peptide, and it might begin at 1010 and 1030 kind of thing. Yeah, and then the same information again. So that is how you get your PTM exchange data back, and you can filter by confidence score, which is useful too. So this brings us to a bit of a close. So further help and documentation, this is all UniProt specific information. So we've got a couple of UniProt webinars coming up. On the 10th of April, we've got an antimicrobial resistance AMR and how we're curating the proteins involved in that. And then on the 9th of May, how we curate and access human disease and protein variant information as well. The webinars I mentioned earlier, we've got a walk through the UniProt website. We've got an introduction to UniProt and how we curate functional information. And the API and Python webinar I mentioned earlier is this one, the programmatic access to UniProt using Python. These are all recorded and freely available at any time. We tend to run them on a pretty annual basis, these kind of webinars. And they're all on I've got a Bitly link here that will give you access to all the UniProt training on the EMBL-EBI sources and the actual UniProt API webinar link here for you. If you want the API access and documentation, that's at this link here. But equally, you can click the API link on the website and find all the documentation for that. If you get stuck doing anything on the website, and I mean anything, you can click the help tabs. They will open help pages that you can navigate. We have a set of frequently asked questions as well. And you can contact us if you need us. So there's always a help bar and a contact us section at the top of the page. There's a help tab that scrolls with you, and there's also a feedback scrolly bar as well. So tell us what you love and what you hate, and someone will always get back to you. I'd like to thank our funding bodies that fund UniProt at all 3 sites. This is the UniProt team. We're broadly split into key staff, content and curation and development teams, which are also broadly spread across the 3 sites around the world. And these are the upcoming EMBL-EBI training sessions. I believe Anna is going to take me back.
Thanks, Emily. So I just popped a comment into the chat to say, please do add your questions to the Q&A box if you have any, and we can have a chat through some of them now. Would you mind just popping back to the previous slide for me?
Of course.
Just while people are having a think about their questions. So Emily mentioned a couple of upcoming UniProt webinars that we'll have in April and May. Those aren't quite ready for registration yet, but they will be soon. So keep an eye out for them on the EBI training site, and also on the front page of the UniProt website. They'll appear there as well. And then I just wanted to highlight a couple of other webinars that we have coming up that are a part of a completely different series, but I thought some people might find this interesting. So all about microbiome, and there's a couple there, but it's a 10-part series, so very busy. But check out the various upcoming webinars there at that link. I know there were various links throughout that were shared, and different ways of doing things. So just to make sure you're all aware that we will share both the slides and the link to everyone tomorrow. So you're very welcome to sort of work back through them, and then obviously get in touch if you need further help, as was mentioned. Okay, so let's have a look to see whether I think maybe in the chat there's a couple of questions coming up. There's an interesting one there for you, Emily. How does one become a protein curator?
Oh, career question. Our backgrounds are very diverse, is a simple answer to that. My background is wet lab. So I did a degree, a master's, a PhD, and then I came to work for EBI. I knew what I was getting into because I'd done an internship between my master's and my PhD doing curation with a GWAS catalog, actually, and they focus on SNP variant annotation. But I worked with them doing some ancestry information, some curation. So when I went to do my PhD, and I realized that, maybe running around a lab isn't what I want. I really enjoyed what I did at EBI. I applied for the protein role as a curator at UniProt. We all have different backgrounds. So because I got the proteomics background, I deal with a lot of the proteomics kind of data that comes into UniProt. But we have specialists doing parasites, different model organisms. We have a Drosophila specialist. We've got a structural annotation specialist. A lot of people have backgrounds where they've done a postdoc or two. Some people have stayed in curation longer than others and have more of a curation background than not. Yeah, it's something you get involved in because you want to do something really different, and you want to provide that information to the community. I think it really appealed to me because I was loving the biology, and you really get to go down into the depth of the biology, but you put it into a resource that's freely available to everyone around the world. Researchers, clinicians, students alike. I do a lot of teaching as well. I go out to a couple of universities and teach with them. I go to conferences still. But I also work a lot with the development team. So there's a lot to it. There's not just putting the data in the database. But yeah, we're all quite broad in our backgrounds and our skill set.
I've also just added a link to a collection of on-demand training we have that's all about biocuration. So that might be an interesting place for you to start if it's something that a route you're looking to go down. Okay, I was just having a look to see. I think Swati's hard at work answering some of the questions, typing away. Somebody asked a question about confidence in tremble sequences. How confident can they be?
So our tremble sequences are all imported. And they're all imported from, annoyingly, I don't have this slide on me. I've set slides, and this isn't this is a different webinar I'm thinking of, but we have a set of resources, Ensemble, ENA, etc., that we import the sequences from. So if they've been, if they're statistically significant and they're confident that they exist in ENA and Ensemble, then they're in our website. So it isn't us that do those particular confidence queries. It would be the other databases, and then we import them. The annotations associated to those imports, as I say, are automatically done, and they're inferred by curator-based rules. So as far as we know, they're accurately annotated on our side before they're then manually curated going forward.
There's also a bit more of a general question. Which is your favorite function or application in UniProt?
Ooh.
That's a tricky one.
I mean, it depends what you want to do. Everyone likes BLAST, I guess, but I like Align. I use Align a lot because I work, at the moment, I'm working on some cardiac proteins, and the big model organism for cardiac proteins is zebrafish. And zebrafish orthologs in mouse and human aren't always straightforward. Some of them are, some of them aren't. So I use a lot of Align, and when you align two proteins together, you can then add all the annotations on. I'm just wondering whether I can show you. I like doing this. Just move that box out of the way. Do I have two interesting queries? Here we go. Oh, look. One I did it. So this is your basket. When I said you can add your proteins to basket, I saved these from earlier. You can click the ones you're interested in, and you can do all your analysis here with them. So I've clicked Align, and it's auto-filled my Align box. I'm not going to name the job. I'm just going to hit go. I'm going to cross my fingers. This one's a real quick one. I like Align because you can add all the annotations onto the sequence if it's got annotations. I mean, I don't know whether these have these. This is an entry I was working on last week, I think, that I've just finished doing. And so all the domains, like as an example, if you've got an active site, you can map that on. So here you've got mouse on top, you've got a zebrafish on bottom. Purple is they're the same and it's conserved. So you see you've got quite a bit of conservation here that moves. And then you can highlight and select annotations. So you can highlight in the mouse certain regions and repeats, or you can even highlight in the zebrafish. Now I know the zebrafish hasn't got that much annotation. There you go. So you can highlight regions. So these are the regions that map to the mouse. And then you can see whether them particular regions are conserved in the zebrafish. And if you've got like an enzyme with an active site and its active site was here, it will be a black kind of spot that you could click on, and it would give you all the information, and it would tell you whether it was conserved between the species as well. And this is really important for things like these proteins. So that this is a protein involved in cardiac development and also in the eye, interestingly enough, because it's involved in vascular development within the embryo. And if these regions aren't necessarily conserved, then are they really doing the same thing in mouse compared to zebrafish? And in this case, this particular protein, no, they're not. It's this particular protein is really important for heart development in chicken, as an example, but not in zebrafish. So I like this feature. I think this feature is really nice. A lot of people use BLAST, and that's worth a good play with. But I just really like Align. There's something very satisfying about Align.
Thank you. There are a couple of questions about the API. It looks like they're being answered via typing at the moment. Someone's also asked in the chat about selecting an organism in the API. And I think that's...
Bring the API up, that's absolutely fine. I said my best piece of advice to the API is to kind of have a look at the website and pick your favorite protein and go down your favorite protein and see where all the information that you're interested in is structured within that entry, and then go to the API, programmatic access, proteins REST API. So this is kind of the home page you're going to land on. And then each one of these sections is openable. So if you hit Proteomics, and then you can search the whole search function. Can I just zoom out a little bit here? There you go. So you can specify species with its tax ID. So I know that's human. I have a feeling that's mouse. So if you only wanted human data, you can put in your tax ID for human or for mouse or for whatever you're looking for. And if you really want, you can bring all peptides associated with that particular species back. You can filter by data source. So if you only want data coming from Proteomics DB or Peptide Atlas, you can filter by that. You can search for your peptide of interest within human or within mouse and make sure that it's unique or not, and things like that. But so that's a proteomics section. But if I was to go to something like features, I'll just open the general one. You see these are slightly different. So your accession ID will go in here. You can search by gene name. You can search by protein name. You can filter by organism name in this case. The tax ID categories there. And then you can look within different categories. So here you got molecule processing, domains and sites, PTM. So everything that's on that feature viewer that is mapped along the amino acid sequence that we've got, you can filter that information here. So if you're looking for something with a modified residue, you can pull back all modified residues within that gene. And you don't even need to filter by species. So you can pull back all the modified residues for beta-catenin across all mammals if you want. But each one of these query formats is slightly different, and you'll find slightly different filters for each bit of information. The key is knowing where to find the piece of information you're interested in. So best thing to do, explore the website, then go to the API and go, right, I know where I'm looking. Now just find my query box and go from there.
Okay, thank you. Looks like the other questions have been answered in the Q&A box. Okay. There is one last question.
Mm-hmm.
And rather than waiting for a typed answer, maybe you could have a quick look. It's quite a long one, so I'm not sure I want to
go for it.
Read the whole thing out. But my proteomic project identifies proteins from the frog Xenopus laevis. As you might expect from Xenopus, functional annotation in UniProt is much less than for human or mouse. Yep. Mm-hmm. However, for each Xenopus protein, there is usually a human ortholog with great annotation in UniProt, often found by a BLAST. Mm-hmm. Is there a quick way, e.g. via the API, to access UniProt entries for the closest human ortholog for a large set of Xenopus proteins?
Ooh.
Could I answer that question quickly? So we do have some proteomics data for frogs. They are from MaxQB, MaxQuant. And we are working with Proteome of Life and Peptide Atlas. And they may provide more frog proteomics data. So we are working on it now. So please watch this space. Yeah.
Well, I think if you were trying to look for a batch of maybe, let's say 1,000 proteins in frog, and you'd wanted to pull back 1,000 human orthologs or near enough. I'm just wondering how you could batch that. Or could you batch that, John? I don't even know. Maybe Swathi knows if we have a perfect ortholog. I don't think we have an ortholog comparison service, do we? As far as I'm aware. Everyone's very quiet. So I don't think we have a perfect ortholog comparison service that I'm aware of. Which I feel your pain because, as I say, I'm doing a lot of zebrafish stuff at the moment, and zebrafish aren't well annotated, but as I say, the human and the mouse are. So I agree with you. It would be a useful feature. But I don't think we actually have a way of looking at that. But all the questions in the chat.
Emily, sorry to disturb. They can use the group by option, group by taxonomy, if to get close to the entries.
Oh, yeah.
In the UniProt website. So if you can share the screen, I don't. We are not sure.
Leo, are you eavesdropping on my webinar? Okay, so where am I going?
Yeah, you can search for your biological question here, right? Yes.
Mhm.
And then on the left side, there is the
by
group by taxonomy. And then it shows it in a, yeah, I don't
Right.
know if that's her question or not. But you can,
Oh yeah, so you could put the query in. Yeah, so you could put the query in, and then you could drill down to groups of. You can either search by mammals or search by frogs. You're going to have a bit of variability between gene names if you do that, but for a rough query, yes, you could do that. Definitely.
yeah.
I don't use that function often, so that's quite useful. Yeah, thank you very much. Or I think you can also, yeah, so you can search specifically by specific taxonomies as well. Or I'm assuming groups, right? Just Xenopus. Yeah. So I don't know if that answers the question, but that's the functionality we've got for that at the moment.
Okay, thank you. I think we're nearing the end of the questions. But I think as we've shown today, there's lots of people in the UniProt team who are around and willing to help answer your questions anytime. So not just today, but obviously, as Emily mentioned before, the contact us on the website is available anytime you have a question and you can ask for some help.
Yep. There's like a little envelope, click that, or there's the feedback bar as well that also gets you to the help desk. And that will then get sent to whoever's the most relevant person to answer your query. We will get back to you.
Excellent. And yeah, as I mentioned before, we will be sharing these slides and the recording tomorrow. So don't worry about all these links. You'll get those to follow shortly. Okay, so I think that just is.
And there's just a very quick question I want to answer. Is why some enzyme lack of false EC number? Our guess is we don't have enough information to determine whether to provide the fourth digit to that EC number, but we try to tell the user, say, we are certain for the first 3 digits, but not the last one. So that happened quite a lot. And the fourth one is the fourth digit for the EC is the substrate difference. So that's a very tricky part.
Yes, as I say, we'll leave it there for today. But any questions in the future, you hopefully know where to go now.